A 2019 study published in Sleep Medicine tracked 847 individuals who consistently slept between 7 and 9 hours per night. 62% reported waking unrefreshed at least 3 mornings per week. Standard sleep architecture looked normal. REM percentages were within range. Slow wave sleep was present. Yet, the subjective experience was fatigue. The researchers documented something the mainstream sleep model does not account for. Sleep quantity and sleep architecture do not predict restorative outcome with the precision we assumed they would. That gap between duration and restoration points to a mechanism operating below the level of sleep stages. It points to what happens inside the cell while you sleep. The textbook model of sleep focuses on cycles, 90-minute intervals, REM versus non-REM, deep sleep percentages. And the assumption built into that model is straightforward. If you complete enough cycles and hit the right stage distribution, you should wake restored. That model works for many people, but for a substantial percentage of the population, it does not. They meet every sleep hygiene recommendation. They avoid screens, they keep the room dark and cool, they get 8 hours, and they wake tired anyway. The mainstream explanation defaults to sleep disorders, apnea, restless leg syndrome, circadian misalignment. Those are real. They account for some of the variance, but they do not account for all of it. And the research over the last decade has been building toward a different explanation. One that reframes sleep not as a behavioral event, but as a metabolic one. In a 2017 paper in Nature Reviews Neuroscience, synthesized evidence from 89 studies examining cellular energy dynamics during sleep. The finding that emerged across those studies was consistent. Sleep is when the brain performs its most energy-intensive maintenance operations. Waste clearance through the glymphatic system, synaptic pruning, mitochondrial repair, protein synthesis. All of these processes require ATP, a lot of it. The brain uses approximately 20% of the body's total energy budget, despite representing only 2% of body mass. During sleep, certain regions of the brain actually increase their metabolic demand compared to waking hours. The restorative functions of sleep are not passive. They are biochemically expensive. And if the cellular machinery responsible for generating that ATP is compromised, and those processes do not complete properly, you get the sleep. You do not get the restoration. That is the architecture. Everything else follows from it. The mechanism centers on mitochondria. These are the organelles responsible for producing ATP through a process called oxidative phosphorylation. Inside the inner mitochondrial membrane, a series of protein complexes transfer electrons down an energy gradient, creating a proton gradient that drives ATP synthesis. Think of it like a hydroelectric dam. The gradient is the pressure. ATP synthase is the turbine. The steeper the gradient, the more efficient the energy production. When that gradient collapses or becomes less efficient, ATP output drops. And the brain is one of the first places you notice it. A 2020 study in cell metabolism measured mitochondrial function in brain tissue across different age groups. By age 40, mitochondrial membrane potential had declined by an average of 31% compared to subjects in their 20s. By age 60, the decline reached 47%. The pattern was consistent across all sampled brain regions. Prefrontal cortex, hippocampus, and brainstem structures involved in arousal and wakefulness. The decline was not pathological, it was normative. And it directly correlates with subjective reports of sleep quality. Here is why that matters for the tired, despite 8 hours problem. During sleep, the brain initiates waste clearance via the glymphatic system. This is a network of channels that flush metabolic byproducts out of the central nervous system. It operates primarily during deep sleep. Cerebrospinal fluid flows through the brain's interstitial spaces, clearing out amyloid beta, tau proteins, and other waste molecules that accumulate during waking hours. A 2018 study in Science demonstrated that glymphatic clearance is an energy-dependent process. It requires functional aquaporin-4 water channels, which are regulated by cellular energy status. When ATP availability is low, aquaporin-4 expression decreases, glymphatic flow slows, waste accumulates. Even if you get deep sleep, the clearance process does not run at full capacity. You wake with a backlog. That backlog manifests as brain fog, sluggish cognition, and the sensation of unrefreshed sleep. The mitochondrial decline also affects synaptic maintenance. During sleep, neurons undergo synaptic pruning. Weak or redundant connections are eliminated. Important ones are strengthened. This process is called synaptic homeostasis, and it is one of the leading theories for why sleep is necessary in the first place. A 2016 paper in Nature Neuroscience showed that synaptic pruning requires coordinated ATP production in both the presynaptic and postsynaptic terminals. When mitochondrial function is impaired, the pruning process becomes less selective. The brain cannot efficiently distinguish which synapses to strengthen and which to eliminate. The result is neural noise, less signal clarity, slower processing speed. You got the sleep, the pruning ran, but the quality of that pruning was suboptimal. And you experience that as cognitive fatigue, even though you were unconscious for 8 hours. There is a second layer to this that the mainstream model underestimates. Mitochondrial function is not static. It responds to metabolic inputs throughout the day. One of the most significant regulators of mitochondrial efficiency is a molecule called nicotinamide adenine dinucleotide, or NAD+. NAD+ is a coenzyme involved in redox reactions throughout cellular metabolism. It is essential for the electron transport chain to function properly. Without sufficient NAD+, the mitochondrial complexes cannot transfer electrons efficiently. The gradient weakens. ATP synthesis drops. And the research published in Cell in 2016 documented that NAD+ levels decline with age at approximately the same rate and trajectory as mitochondrial function. By age 50, NAD+ concentrations in human tissue are roughly half of what they were at age 20. The decline is steepest in high energy demand tissues: brain, heart, skeletal muscle, liver. And NAD+ levels follow a circadian rhythm. They peak during the day and decline at night, which means the brain enters its highest metabolic demand period, sleep, at the point when NAD+ availability is lowest. If baseline NAD+ is already depleted due to age or metabolic stress, the brain does not have the biochemical resources to complete its maintenance protocols. The processes run in degraded mode. That is not a metaphor, that is the mechanism. Now, here is where the pharmaceutical model intersects with this biology in ways most people do not realize. Statins are among the most commonly prescribed medications in the United States. Over 40 million adults take them. Statins inhibit an enzyme called HMG-CoA reductase, which blocks cholesterol synthesis. That is the intended effect. The unintended effect is that the same biochemical pathway produces coenzyme Q10 or CoQ10. CoQ10 is a critical component of the mitochondrial electron transport chain. It shuttles electrons between complex 1 and complex 3. Without adequate CoQ10, the entire chain slows down. A 2015 meta-analysis in the American Journal of Cardiology reviewed 17 trials involving statin users. Every trial documented measurable CoQ10 depletion. The average decline was 32% after 12 weeks of statin therapy, and multiple studies within that meta-analysis found a significant association between CoQ10 depletion and subjective reports of fatigue, even in patients whose cholesterol numbers improved. The cardiovascular benefit was real. The mitochondrial cost was also real, and because CoQ10 depletion affects the brain just as much as it affects muscle tissue. Statin users frequently report sleep that feels less restorative. And even when objective polysomnography shows no structural abnormalities, the sleep stages are intact. The cellular energy production is not. All of this points to one place. The restorative quality of sleep is not determined solely by sleep architecture. It is determined by whether the brain has the metabolic capacity to execute its maintenance operations during the time it is offline. Duration matters. Architecture matters. But cellular energy availability is the limiting factor, and that factor operates independently of how many hours you spend unconscious. Research has identified a natural compound that supports this neural and mitochondrial mechanism at the cellular level. It is not a drug. It is not a treatment. It is a natural formula built from published research on brain and cellular energy metabolism. And if you want to understand how it works, the link is in the description. This is a sponsored recommendation and individual results vary. The practical implication here is not that sleep duration is irrelevant. It is that duration alone is not a sufficient framework for understanding restorative sleep. The labs measure disease. This mechanism operates below that threshold. Standard of care is built around pathology. Sleep apnea, insomnia, circadian rhythm disorders, those are real conditions with real interventions. But what we are describing is optimization, and those are two different frameworks. Pathology asks, is something broken? Optimization asks, is the system running at full capacity? Most people who wake tired after 8 hours do not have a diagnosable sleep disorder. They have a system running in degraded mode. You know, the difference matters because the interventions are different. Pathology gets a CP, AP machine. Optimization gets mitochondrial support. One of the more useful distinctions that emerges from this research is the difference between sleep debt and metabolic debt. Sleep debt is the model most people are familiar with. You miss sleep, you accumulate a deficit, you repay it by sleeping more. That model works for acute sleep deprivation. It does not work for the chronic unrefreshed sleep we are discussing here. A 2018 study in the Journal of Clinical Sleep Medicine followed subjects who reported persistent fatigue despite adequate sleep duration. Researchers had them extend their sleep by 90 minutes per night for 2 weeks. Total sleep time increased. Subjective fatigue did not. The sleep debt was repaid, but the fatigue persisted. The researchers concluded that the fatigue was not a function of insufficient sleep quantity. It was a function of insufficient metabolic recovery during the sleep that was already occurring. Metabolic debt is not repaid by sleeping longer. It is repaid by improving the efficiency of the cellular processes that occur while you sleep. That is a fundamentally different intervention point. Here is the second order implication that most discussions of sleep miss. If mitochondrial function determines restorative capacity, then anything that impairs mitochondrial function during the day will affect sleep quality that night. Chronic stress, insulin resistance, systemic inflammation, nutrient deficiencies, all of these degrade mitochondrial efficiency in real time. A 2019 paper in Frontiers in Neuroscience demonstrated that acute psychological stress reduces mitochondrial membrane potential within hours. The effect is measurable and reproducible. The mechanism involves cortisol-mediated disruption of calcium handling in mitochondria, which impairs the electron transport chain. If you spend your waking hours in a state of elevated cortisol, You are entering sleep with compromised mitochondrial function. The restorative processes will not run at full capacity. You will sleep 8 hours, you will wake tired, and no amount of sleep hygiene will fix it because the problem is not sleep. The problem is daytime metabolic stress bleeding into nighttime recovery capacity. The system is connected. You cannot optimize one end without addressing the other. That framework also explains why some interventions work for some people and not others. Magnesium supplementation improves sleep quality in individuals with magnesium deficiency because magnesium is a cofactor in over 300 enzymatic reactions, including several steps in ATP synthesis. If you are deficient, supplementation restores mitochondrial function. If you are not deficient, supplementation does nothing. The same principle applies to CoQ10, B vitamins, and other micronutrients involved in energy metabolism. The intervention works when it addresses the actual metabolic bottleneck. It fails when the bottleneck is elsewhere. And for many people, waking tired despite adequate sleep, the bottleneck is not a single nutrient. It is cumulative mitochondrial decline driven by age, stress, and metabolic load. That is a systems-level problem, not a deficiency problem, and it requires a systems-level approach. If this gave you a new way to think about your biology, leave a comment below. I read every one. And if you found this useful, the single best thing you can do is follow the channel. We decode a new mechanism every day. Everything you need is in the description and in the first comment. And here's the practical question that framework creates. How do you measure mitochondrial function in your own body? Standard lab work does not do it. You can get a complete metabolic panel, thyroid function, vitamin D, iron, B12. All of those are worth checking, but none of them directly measure ATP production capacity. The closest proxy available in clinical practice is lactate. When mitochondria cannot produce ATP efficiently, cells shift toward anaerobic glycolysis. That produces lactate. Elevated resting lactate, particularly if it is above 1.5 millimoles per liter without recent exercise, suggests mitochondrial inefficiency. It is not definitive, but it is directional. The other accessible marker is heart rate variability, or HRV. HRV reflects autonomic nervous system balance. An autonomic function is tightly coupled to cellular energy status. Low HRV, particularly during sleep, correlates with reduced mitochondrial capacity. If your HRV is consistently low despite adequate sleep duration, that is a signal, not a diagnosis. A signal, and it suggests the system is running in a compensated state. Before you go, we built a free 2-minute biological wellness assessment that reviews 4 body systems and gives you a personal pattern score. The link is in the bio, in the first comment, and in the description. 2 minutes. It puts numbers to everything we just discussed. The next mechanism we decode, why your cognitive performance drops in the afternoon even when your sleep was perfect. The final layer that most people miss is the interaction between mitochondrial function and circadian biology. The circadian clock is not just about when you feel sleepy. It is a master regulator of cellular metabolism. A 2021 study in Cell Metabolism demonstrated that mitochondrial efficiency follows a circadian rhythm. ATP production capacity peaks in the early afternoon and reaches its lowest point in the early morning hours. That rhythm is controlled by clock genes that regulate the expression of mitochondrial enzymes. When circadian rhythm is disrupted, shift work, jet lag, and inconsistent sleep-wake times, the circadian regulation of mitochondrial function becomes desynchronized. You get mitochondria trying to run maintenance operations at the wrong time in the metabolic cycle. The brain expects high ATP availability during sleep. The mitochondria are not delivering it on schedule. Even if you eventually fall asleep and get 8 hours, the timing mismatch reduces the efficiency of every ATP-dependent process. The glymphatic system runs slower. Synaptic pruning is less precise. Protein synthesis is incomplete. You meet the duration threshold, you miss the restoration threshold, and you wake tired. This is why some people feel worse after sleeping in on weekends. They assume more sleep equals more recovery. But if that sleep occurs at a circadian time, when mitochondrial function is downregulated, the recovery is suboptimal. A 2020 paper in Sleep Health analyzed data from over 1,200 subjects using actigraphy and cognitive testing. Subjects who maintained consistent sleep-wake times within a 30-minute window across all 7 days of the week reported higher subjective sleep quality and performed better on cognitive tests than subjects who extended sleep on weekends but shifted their sleep timing by more than 90 minutes. Total sleep time was actually higher in the weekend shift group. Cognitive performance was lower. The circadian disruption cost more than the extra sleep gained. The system prioritizes timing over duration when those two variables are in conflict. The pharmaceutical industry has known about the mitochondrial dimension of sleep for longer than the public realizes. Certain classes of psychiatric medications, particularly atypical antipsychotics and some SSRIs, are documented to impair mitochondrial function. A 2017 review in Psychopharmacology examined mitochondrial effects of commonly prescribed psychotropic medications. Multiple drug classes showed measurable reductions in mitochondrial membrane potential, CoQ10 levels, or ATP synthesis capacity. The magnitude of effect varied by drug, but the pattern was consistent. And one of the most common side effects reported across those drug classes is fatigue and unrefreshing sleep. Not insomnia. The patients sleep. They just do not wake restored. The clinical literature attributes this to sedation, but sedation and restorative sleep are not the same process. Sedation suppresses arousal. Restoration requires functional cellular metabolism. Now, if a medication is impairing mitochondrial function, it is directly undermining the metabolic processes that make sleep restorative, regardless of how many hours the patient spends unconscious. That is not an argument against psychiatric medication. It is an argument for understanding the metabolic cost of those medications and considering whether mitochondrial support should be part of the treatment protocol for patients on long-term pharmacotherapy. The research that ties this all together was published in 2022 in Nature Aging. Researchers used a technique called phosphorus magnetic resonance spectroscopy to measure brain ATP levels in living subjects across different age groups. They found that ATP concentration in the prefrontal cortex declined by an average of 19% per decade after age 30. The decline was steeper in individuals who reported chronic sleep disturbances. And here is the critical finding. When researchers compared subjective sleep quality ratings to objective ATP measurements, ATP levels predicted sleep quality more accurately than any polysomnography variable. Not REM percentage, not slow wave sleep duration, ATP. The cellular energy reserve available to the brain at the start of the sleep period was the strongest predictor of whether subjects would wake feeling restored. That finding reframes the entire conversation. Sleep is not just a neurological event, it is a metabolic event, and the quality of that event depends on whether the cells executing it have the energy to do the job.